So, hello and good evening, all of you. Welcome to the live session of week 11. I am Surasree, and in today's session, we will be discussing autoimmunity and transplantation. Okay, so to answer your doubt, no, today is not the last class. Today is week 11. I have one more class with you. Okay, so you're asking like in which way the release of calcium from the ER causes the release of the histamines. Okay, if I have this previous slide. Yes. Okay. So the release of calcium. So I told you that the PKC, the protein kinase C, and also the role of IP3 to release calcium from the endoplasmic reticulum, right? And if this calcium is released, then what it helps in? It helps in the contraction of the microtubules inside the cell. So these are the granules that contain these pro-inflammatory the pro-inflammatory molecules, right? Like for example, histamine or prostaglandin. So these granules are within the cell, and if these granules have to be exocytosed, right? So if these granules have to come out of the cell, then these granules have to fuse with the cell membrane, and then the contents of these granules will come out of the cell membrane, right? So for that we need the help of the microtubules. So you know that microtubules are the roadworks, are kind of the roadworks inside the cell. So like it creates the path by which the granules will move towards the cell membrane and will ultimately be fused with the cell membrane, right? So that is the role of this calcium. So it helps in this contraction of the microtubules. Is that okay? Okay. All right. So let's begin today's lecture then. Anyone having any other questions from previous class? All right. If not, then let's begin today. So today we will be discussing autoimmunity and transplantation. So what is autoimmunity? So autoimmunity, by the name you understand that it is immunity against the self, right? Or auto means usually self. So the immunity against the self. So while we were discussing the T cell and B cell maturations, so we discussed that the T cells and the B cells, the lymphocytes, they undergo several rounds of selection when they are developing and when they are maturing, right? And in that context, the T cells and the B cells, which can recognize the self antigens, that means which can elicit an immune response against the self antigens, they are usually deleted, or they are treated in such a way that they cannot be active. So either they are made unresponsive, or they like the immune system ignored these population of cells which act against the self antigens, right? However, there are several cases where these tolerance mechanisms means the mechanisms which prevent this kind of self reaction, these tolerance mechanisms can be breached in some cases, and in those cases what happened that our own T cells and B cells, they can elicit an immune response against our self antigens, and that causes the autoimmune diseases. And if we look at this table, we will see that the autoimmune diseases are not that rare also. Like for example, you can see here in case of psoriasis, so it is a most common, it's a very prevalent autoimmune disease, and this is something that we can see in 1 in 50 people, and it forms kind of patches on the skin, and because of that what happens, it's like it forms this plaques kind of things. And why does these appear? Here you can see so the disease, the mechanism of the disease and the consequence, and also the prevalence is listed in here. And you can see that the autoreactive T cells, when, so autoreactive means what? They are reactive against the self antigen. So the autoreactive T cells, they act against the, or they recognize the some skin associated antigens or some skin associated proteins or molecules, because of which it causes the inflammation of the skin and cause this disease psoriasis. Whereas another very common autoimmune disease is rheumatoid arthritis, also myasthenia gravis. So what are these diseases? For example, rheumatoid arthritis is when the T cells and the antibodies, they act or they recognize the antigens which are located to the joint synovium, right? The joint fluid, the synovial fluid of the joint, which are responsible for lubrication of the joint. When the T cells and the antibodies, they act against the tissues, against this joint of the joint synovium, and it causes the joint inflammation, and finally it causes arthritis. So this is rheumatoid arthritis, which is also a very common autoimmune disease. You can see in almost one case in 100 people. And then myasthenia gravis or Graves disease. Graves disease is actually when there are autoantibodies. So there are antibodies which are produced which act against the thyroid stimulating hormone receptor. And obviously, since the thyroid stimulating hormone receptor is is being acted upon by these autoantibodies, therefore it causes hyperthyroidism. It is also, you can also find it in 1 in 100 people. And another common disease is systemic lupus erythematosus. So SLE or SLE. SLE is a disease when the T cells act against the DNA, the chromatin proteins, and also the ubiquitous ribonucleoprotein antigens, right? So it causes a systemic disease, that is why it is called systemic lupus, and it causes vasculitis, rash, and glomerulonephritis. So it causes inflammation of the glomerulus inside the kidney, right? So these are some of the Crohn's disease, multiple sclerosis, and these are some of the autoimmune diseases that are commonly available, that can be seen in people. And obviously from these prevalence, you can understand that in several people, this tolerance mechanism, this tolerance that ensures that your self antibodies or your self T cells will not act against any self antigen, this tolerance mechanism is breached. So while I'm talking about tolerance mechanism, let's just discuss what are these tolerance mechanisms. So tolerance mechanism can be of two types. One is central tolerance, another is peripheral tolerance. Central tolerance and peripheral tolerance. So what is central tolerance? Central tolerance means that when the T cells and B cells, when they are developing in the thymus or the bone marrow, right, before they reach out to the peripheral lymphoid organs, right, before they go to the peripheral lymphoid organs, they are maturing in the thymus or the bone marrow. There, the kind of tolerance mechanisms that are programmed in these T and B cells, that is called central tolerance. Okay. So central tolerance obviously involves deletion and editing of the B cell and T cell receptors. Right? Deletion means what? Suppose that the T cell is reactive against the self antigen, then what is done? The T cell die by apoptosis, or it is made to die, right? So that this particular clone of T cells are deleted or B cells are deleted. or editing, like when the T cell and B cells, they undergo this receptor editing. In that phase, if that particular receptor is being reactive against self antigen, then this receptor is edited multiple times so that this receptor cannot act against self antigens. So this is one mechanism, that is central tolerance. Now coming to the several peripheral tolerance mechanisms. Peripheral tolerance means that which happens, the tolerance that happens during the time the T cells and B cells, they reach the peripheral lymphoid organs, right, after they leave the thymus or after they leave the bone marrow. There are also some tolerance mechanisms still undergo, right, so that selection of the T cell and B cell repertoire doesn't finish in your thymus or bone marrow, right, so it goes on continuously. So for example, you can see that antigen segregation. Antigen segregation is what? So first of all, there's a physical barrier which actually segregates self antigens to access to the lymphoid system. So this is nothing but just a physical barrier that the self antigens cannot come in the lymphoid system, and it cannot stimulate any T cell or B cell. Then peripheral anergy. So anergy is one term that we had mentioned in while we were discussing T cell B cell maturation also. Anergy is something which inactivates the cells, the T cell or B cells, which act against the self antigens by weak signaling. So they do not receive the co-stimulus which are required for their differentiation and activation. So this happens in the secondary lymphoid tissues. Next is the regulatory T cells. Regulatory T cell is one important component of the immune system, which what does it do? It inhibits any kind of immune response, right? And what it does is it suppresses the production of cytokines, and it causes some intercellular signals which regulate or which inhibit the production of several types of T cells. So it also occurs in secondary lymphoid tissue and also in the sites of inflammation. Next is functional deviation. Functional deviation is one important concept where the T cells that produce the inflammatory cytokines, they are differentiated to produce regulatory T cells or Treg cells, right? So suppose one particular T cell is producing it is acting against the self antigen, and it is able to produce inflammatory cytokines. So obviously it will cause inflammation and autoimmune disease. So this T cell, if it is now programmed to differentiate into Treg cells, then it will cause inhibition of this inflammation, right? So this functional deviation also takes place. And the last one is activation induced cell death, which is nothing but apoptosis. So the cells are died, so the cells die by apoptosis if it is reactive against self antigens, right? So these all tolerance mechanisms work, and one concept here I would like to discuss, which also comes under central tolerance, that is the role of a particular gene which is called autoimmune regulator, or it is in short form it is known as AIRE. So what is the role of this autoimmune regulator? It promotes the expression of some tissue specific antigens in the thymic medullary cells. What does it mean? So our different tissues express different self antigens, right? Or different proteins, different antigens are expressed in different tissues of our own body. Whereas you know that the central tolerance, it occurs in the thymus only, right? Thymus or the bone marrow. So therefore the T cells that will come out of the thymus, they are required to not act or not recognize the self antigens of the eye, the self antigens of the ovaries, the self antigen of the liver, the self antigen of the intestine, right? Because all these self antigens, all these antigens should, like the T cell population should not react against all these antigens, right? So therefore they have to recognize that these are the self antigens, right? But how does this recognition take place? Because the T cells are maturing in the thymus only, Therefore, if one particular protein is being expressed only in the eye or only in the uterus, then how is this protein, like how does this T cell recognize that yes, this is a particular protein which is expressed in the eye, and I will not go and react against it? How does this recognition take place? So that is why there is this particular gene which is very interesting, which is the autoimmune regulator, and it expresses some tissue specific antigens in the thymic medullary cells, right? So this is a particular gene which helps in expression of some tissue specific antigens in the thymic cells, so that the thymus can express these particular antigens to the developing T cells there. Okay, so here you can see that the individual organs, they express some tissue specific antigens, but in the thymus the T cells arise which are capable of recognizing tissue specific antigens because of this gene which is called AIRE, right? So under control of this AIRE protein, the thymic medullary cells express tissue specific proteins which lead to the deletion of these tissue reactive T cells, right? So since they are now expressing these tissue specific proteins, now the T cells can recognize that these are the self proteins, and therefore they will not react against these self proteins, right? So if they react, they will be deleted. So this control is present in the thymus also, right? And also what is observed, that in the absence of AIRE, which was observed in mice, that the T cells which are reactive to tissue specific antigens, they mature and they leave the thymus, and of course they can also cause these autoimmune diseases, right? So is that clear, the role of AIRE so far? Any questions? Okay. Okay, so now I will not go in details of the autoimmune diseases. I hope you can find them from the lectures, but yes, Vandana?
I have a query.
Yes.
Like for the autoimmune disorders, there are for like tissue transplantation, there are some drugs. Sir did not go into the details of the drugs, and she said it is not in the scope of the course. But he said that the drugs, so many newer drugs have developed. Even if it is not taken from the close relative, the transplant can be. There are more
Yes.
chances that the transplant is not rejected.
Yes.
So, in the same, aren't there any drugs for these autoimmune disorders also?
Actually, it is quite difficult because, you know, like in case of transplantation, what happens, there are some immunosuppressive, these drugs are called immunosuppressive drugs, which are used so that this immune reaction is not elicited against the cell, the antigens or of the graft, right? Or against the graft. But autoimmune disorders are a little difficult to treat, but there are some drugs which can actually exacerbate or which can actually reduce the effects of the symptoms. I think those drugs are there, but like I will also need more information about that to tell you in detail.
Thank you. Thank you so much, ma'am. Thank you.
Okay, as far as I know that.
Can I say a few words, ma'am?
Yeah, please go on.
Yeah, as of now, I know that for psoriasis, okay, which is an autoimmune disease
Yes.
again, for that they are prescribing, immunosuppressive drugs in the form of creams.
Right.
Okay, so it is being given, Vandana madam.
Thank you so much, ma'am. Thank you.
So for psoriasis, at least I know that yes, there is tacrolimus cream which is available and which can treat psoriasis maybe when it is in a minor stage or something like that.
Uh, do you know, Nisha, like which path or which pathway that drug is working to help this autoimmune?
Tacrolimus, as sir had mentioned, okay, he had mentioned various pathways, okay, by which the drugs are acting.
So
Okay,
these
so in that,
pathways,
I think cyclosporin and tacrolimus was mentioned in the same pathway. I don't remember which pathway, but
I think it was related to cell division, ma'am. I think it was related
actually
to
I
yeah.
cell
So
have
in
that
that
division.
slide.
picture, he has mentioned the specific pathway, sir.
Yes, I have that slide where I'll be discussing also about these drugs. So that will clarify your questions, Sandeep.
Okay.
Okay, so any other doubts till this, or I will move on to the next part, which is the transplantation. Okay, all right. So moving on to transplantation, first I would like to just describe two or three terms here, which are allograft, autograft, xenograft, and syngeneic graft. Can anyone give me an idea about what are these different types of grafts? Yes, Vandana, go on.
Ma'am, allograft is when a tissue is or organ is planted from one individual to the other of the same species. Autograft is, say, for example, the skin is transplanted from feet to the face, like in the same individual.
Exactly.
And xenograft is when the tissue or organ is transplanted in between two different species. And syngeneic graft is that like is similar to the isograft, that is transplanted in between two identical twins.
Exactly. Very good. So allograft and syngeneic graft, these two are like this is happening between two individuals of the same species. Allograft and syngeneic graft both. But in case of allograft, the grafting is happening between two individuals of the same species but who are not genetically identical, right? However, the syngeneic graft is occurring, it is a transplantation process, transplantation of cells or tissues or organs between two genetically identical persons who might be the identical twins, right? Otherwise, that's not possible. Like any individual is not genetically identical to any other person, right? So these are the different types of transplantations that are actually possible. So this thing that autograft is the grafting, the transplantation between different sites of the same animal or person. So therefore we know that autografts are usually 100% successful, because if we are transplanting one part of, suppose, a skin from my arms to skin from my neck, then of course that does not make any difference to the immune system. Like immune system knows that this is only my proteins and my self antigens, so therefore it does not react. However, um, when the skin is grafted between two unrelated individuals, that means two different individuals of the same species or allogenic individuals, then the graft initially survives. So the transplantation, it initially survives, but later on it is rejected in about 10 to 13 days after the transplantation. So this response is called an acute response. It's called an acute rejection of the graft, and we will know that this occurs because of the T cells, right? And how do we know that it is because of the T cells? So there were several experiments which were carried out, and it was observed that when the grafting occurs between so the skin from one mouse is grafted onto a nude mice. So because of the nude mouse, they do not have the T cells, right? Because they do not have the thymus. So when this grafting occurs onto the nude mouse, then this kind of rejection does not happen. So of course from that it was discovered that it is the ability of the T cells to elicit a response towards the graft or the transplanted organ or tissue, right? So so what happens here, you can see that when the skin graft is done to a syngenic recipient, that means genetically identical. So this MHC, so MHCs have these haplotypes. Do you remember? So it's the particular MHC molecules that are being expressed in one particular individual, right? So I told you that MHCs are polymorphic proteins, right? So they are like in any two individuals, the MHCs are going to be different. So therefore, if a skin or something, any tissue or anything is grafted onto a syngenic recipient who have the same MHC haplotype, then the graft is tolerated, right? Then the graft is tolerated and all the grafts survive. However, if skin is grafted onto an allogenic recipient, which is not genetically identical. So you can see this yellow mouse, it has MHC A, whereas this blue mouse has MHC B. So when this MHC A from the yellow mouse is transplanted to the tissue of this yellow mouse is transplanted to the blue mouse, then what happens, that this there will be T cells which will recognize this MHC A as a foreign antigen, and then it will react against this particular transplant, right? So the graft is rejected rapidly, okay, after 10 to 13 days. However, if you next what you do is, if you do a second skin graft from the same donor to the same recipient, so again you are taking a part of the skin from the yellow mouse and you are transplanting it onto the blue mouse, then what happens, this graft shows accelerated rejection. Okay, so the second time the response is much faster, right? And when the T cells are transferred accelerated rejection from a sensitized donor to a naive recipient. So suppose this particular mouse, okay, so from this particular yellow mouse, the part of skin is being transferred to a naive mouse, to a naive blue mouse. But if this MHCB, which is sensitized to MHCA, if it is transferred, so these are two different mice actually. So this is one and this is two. Okay, this is one. So what is done? From this particular yellow mouse, I am transferring a graft onto this blue mouse, and also I am transferring this MHC molecules from this first mouse to the second mouse. First mouse is already sensitized, so it has already shown this graft rejection. Then what happens, we show that the graft is rejected much faster. So this is a first time when you transplant.
Ma'am, T cells are transferred from the sensitized mice to the naive mice.
Exactly. T cells are transferred. Sorry, I accidentally mentioned about MHC, not that. T cells, when they are transferred from this mouse 1 to mouse 2, then they show this accelerated rejection, which is much like the second set rejection, like when the same mouse is again transferred from the first mouse, right? So this kind of response can be observed. This rapid course of second set rejection can also be observed because these T cells are now primed for the donor skin. They can recognize that yes, there has been a previous infection, or there has been a previous recognition from this particular from this different MHC type, right? So this is what happens when as the T cells are involved in this anti-graft response. Now I'm coming to one particular protein which are called minor H antigens. Now what happens is that, so since these MHC molecules, they generate this kind of they are responsible for this response against the transplants. So that is why, so before any transplant, MHCs are checked. Okay, so MHCs are checked in between two individuals, between the donor and the recipient, and even if the MHC molecules are identical between two individuals, then also we can observe a anti-graft response or a rejection of the graft. Now, how does that happen? I am telling you that the MHC molecules are responsible because the T cells of the recipient, they act against the MHC molecules of the donor, which they recognize as the foreign. But if the MHC molecules, they are identical, then how does this response again originate between two identical MHC person, right? Between two identical MHC individuals. That actually is due to the presence of these minor H antigens or minor histocompatibility antigens. So what are these minor H antigens? They are peptides which are derived from polymorphic cellular proteins. So some proteins of our body, not only the MHC proteins, the major histocompatibility complex proteins, but also some other proteins are polymorphic in our body. That means your protein will be different from mine, or so the proteins differ, the amino acid sequence differs between individual to individual, even if it is the same protein, right? So this kind of polymorphicity exists in any population, and because of that, these proteins, they are the peptides which are derived from these polymorphic cellular proteins, they bind to the class 1 MHC molecules, and these self proteins are digested by the proteasomes in our cells. You know that any proteins are digested by the proteasomes, and then the peptides that are delivered from them, that are derived from them, they are delivered to the endoplasmic reticulum, where they bind to the MHC class 1 molecules, and then finally they are delivered to the cell surface. Now what happens if this polymorphic protein, if it differs between the donor and the recipient, then it can give rise to an antigenic response, which can be like this particular antigenic peptide can be recognized by the recipient's T cells as foreign, right? Because of these polymorphic proteins. So here you can see that suppose this red protein and the blue protein, these are actually polymorphic proteins. So they are same proteins, but in the donor and the recipient, they have slightly different amino acid sequences, right? So because of that, what happens, this red protein and the blue protein, when they will be expressed on their cell surfaces, right, they will be recognized as different antigenic peptides, right? So therefore, in this recipient, this particular red antigen bound to this particular MHC will be recognized as foreign, and therefore it will the recipient's T cells will launch an immune response against this donor's polymorphic proteins. So this is the role of minor H antigens, which is why even if in the MHC identical persons, between the MHC identical donor and recipient also, we need to apply or we need to treat the recipient with some immunosuppressive drugs before the transplantation can take place. right? Because even if we check that the MHC, the major histocompatibility complex, these proteins are completely identical in these two individuals, the donor and the recipient, then also these minor H antigens will be present, against which there will be some immune response which will be launched or which will be elicited. So therefore there is the need of these immunosuppressive drugs. Now I will come to the immunosuppressive drugs later on. So these are the minor H antigens, right? And the response to these minor histocompatibility antigens is very similar to what happens in case of viral infections. So whereas any antiviral response eliminates only infected cells, but what happens in these minor H antigens, the graft is totally destroyed in response to these minor H antigens, right? So that causes the rejection of the graft. Now we will talk about the allo antigens and the alloreactive T cells. So what are these allo antigens? These are the antigens that differ between the members of the same species, right? So these are the allo antigens, and the immune response against such antigens is known as alloreactive response. Now this alloreactive response or the allo recognition creates or initiates this acute graft rejection. So I was mentioning this in the beginning that within 10 to 13 days it can be observed that this graft is totally rejected, right? How does this happen? So here you can see that the kidney is transplanted, right? And the kidney contains the dendritic cell of the donor, right? Now these dendritic cells, they migrate to the lymph node. After this kidney is transplanted to the recipient, these dendritic cells migrate to the lymph node and the spleen via blood, and they activate these effector T cells. So how does this activation take place? So these dendritic cells, these dendritic cells, they express these MHC molecules, right, which are its own MHC molecules, and also they display some antigenic peptides on its surface, right? So these are the tissue resident dendritic cells. Now these dendritic cells, when they go to the lymph node and they via blood, now when they reach the lymph node, the effector T cells of the recipient, they can recognize this complex, this particular MHC peptide complex, as foreign, and then they become activated, right? So this is recognized as foreign by recipient T cells, and then these T cells, they mature, they are activated, they are differentiated, and now these effector T cells, they migrate to the graft, because this particular dendritic cells are present in the graft, so they migrate to the graft now, and then the graft is destroyed with the action of these effector T cells. So this is called an acute rejection, when the recipient's T cells, they recognize the complexes of the donor HLA or the donor MHC along with the donor peptides. Right?
But how it is totally destroyed, like whole kidney cell will be destroyed, is it?
Yes, the whole graft is actually rejected. So all these T cells, they now migrate to the graft, and as I mentioned that this is not something like that the only the T cell, only the cells that are expressing this particular complex of, which I was writing, complex of donor MHC, donor peptide, only those cells will not be destroyed. Rather, all the cells that are present in the graft, they will be killed.
Like how then they will identify, like how the antibody will identify those other, or the T cells will identify the other cells?
Actually, it creates a cytokine milieu where these cytokines actually act on the nearby cells also. So, in that way, this whole graft, and this is not very clearly like figured out, but the whole graft is actually rejected. Like all the cells are rejected, and the graft is
Okay.
destroyed.
So, but is there any way, like before transplantation, we can verify, so that like why to wait to transplant, then cost include, and then wait for 10 12 days,
Exactly,
and
exactly.
then
That
reject?
is what I mentioned. That is why these MHC molecules are checked whether they are identical or not. But even if the MHC molecules can be totally identical, then also there will be some minor H antigens against which this response can be generated. Yes, Vandana.
I need to add here that all the grafts are going to be rejected, whether there is HLA matching done or not. All the grafts will be rejected. To prevent this rejection, immunosuppressive drugs are given.
Exactly.
Only because of those immunosuppressive drugs, the rejection can be reduced.
Exactly.
The chances
Then
of
also,
rejection
then
can be
also,
reduced.
then
And
also.
I had a query, ma'am.
Yes, yes.
These dendritic cells that are coming from the donors, the MHC molecules we are talking about, are they MHC1 or 2?
Both. Both.
Both.
Yes.
Okay, ma'am.
Both MHC1 and 2.
Yeah,
Thank
I
you so
think
much.
because dendritic cell express all types, right? MSC1, MSC2, everything. Right.
Yes,
So
yes,
that's
yes.
why
Yes,
it
yes.
can.
Yes. Okay, so this is called acute graft rejection. Okay, it occurs 10 to 13 days post the transplantation, right?
May I say something out here, ma'am?
Yeah, sure.
Yeah, I think the blood supply is stopped near the arteries, like at the junction, and that's why it is again rejected.
Yes, that also happened, and there is one thing which is called a chronic rejection, where this particular thing happens that the blood supply, the arteries, they are actually choked. So
Okay.
it is not acute like as fast as acute, but after some time, this kind of chronic rejection develops, which ultimately leads to the rejection of the graft, but occurs much later.
Like this can happen with the minor H antigen
Yes,
cases.
yes,
Okay.
yes.
It was covered, ma'am. It was covered that it would lead
Exactly.
to uh, innate immune response will be active then.
Exactly.
Through the HEVs, the immune
The
cells
cells
will
will
go and that
migrate
will
to the
cause
blood
hemolysis.
vessels, and
It
there.
takes roughly around 60 days.
Exactly. So after some time, this will be finally rejected, but it takes much later, much longer time than acute, or as I will next mention, hyperacute response. Okay. So this is acute graft rejection. Now moving on to hyperacute graft rejection. So hyperacute, obviously from the name you understand that the graft failure occurs much faster than than acute graft rejection. So this is hyper, and why is it taking much lesser time? Because it is caused by the antibodies as well. All right. So this pre-existing alloantibodies against any blood group antigens or any polymorphic antigens, polymorphic MHC antigens, can cause this rapid rejection of transplanted organs. So if the antibodies are present in the recipient from before, you understand that if, so for example, this occurs against the antibodies of the blood or the blood antigens. The blood contains several antigens, right? The Rh antigens, for example, or the other ABO antigens. So if the blood group is not matched between the donor and the recipient, then what may happen is that the because of some previous exposure or some previous maybe blood transfusion or anything, the antibodies against those particular blood antigens might be present in the recipient from before, And now if the transplant is actually done, if the organ is transplanted, then what happens, these antibodies will quickly come and destroy the graft. So it will create graft, it will generate graft failure much faster. Okay, so here you can see that the healthy kidney which is grafted on into the patient with kidney failure, but there are pre-existing antibodies against donor blood group antigens in the recipient, right? So then what happens, these antibodies, they bind to the vascular endothelium of the graft and it initiates an inflammatory response. So this inflammatory response, they occlude these blood vessels, right? And then the graft, this becomes purple colored and hemorrhage begins, which ultimately leads to the failure of the graft or the rejection of the graft. All right, so it causes thrombosis of the graft and it causes deoxygenation, so therefore it leads to rejection of the graft. So this problem can be avoided by the ABO blood group matching, also the Rh antigen matching between the donor and the recipient. Okay, also there is cross matching. Cross matching involves determining whether the recipient has antibodies against the white blood cells of the donor. That is also checked to avoid this kind of hyperacute graft rejection. Is that okay? Vandana, you want to say something?
Ma'am, I have a query. Like sir said, even the fetus will be having two different haplotypes, and the mother will have
Exactly.
mother will treat half of those haplotypes coming from father as foreign. So
Exactly.
is it possible that because of conception and after the delivery, maybe when if mother requires any organ and that is given by the father, so will that show acute graft rejection because of this previous exposure through fetus?
Might be possible. Yes.
Okay, thank you.
Yeah. Okay. All right. Moving on. Anyone having any doubt still so far? Okay. If not, then moving on to the immunosuppressive drugs that we were discussing from the beginning. The requirement of the immunosuppressive drugs, so far we have understood that even if there is matching or cross matching or the matching of the blood group antigens, then also there is some possibility that some antigens will be treated in our body by, like some antigens of the graft will be treated as foreign and an immune response will be generated against the foreign antigens. So to actually to avoid that, we need the help of some immunosuppressive drugs which are now available due to the improvement of our medical technologies, and these are some of the drugs that I'm going to discuss. One is alemtuzumab. This one is alemtuzumab. This is a monoclonal antibody against CD52. Okay, and what it does is that it is used to deplete T cells before transplantation. So these immunosuppressive drugs are given to the recipient, right? Next is this anti-CD3 monoclonal antibody acting against CD3. What it does is that it prevents the generation of signaling by the CD3. Okay, so CD3 is responsible for signaling of the TCR complex. So anti-CD3 monoclonal antibody inhibits this signaling by the TCR complex. So that is how it inhibits also the T cell response. Next is this cyclosporin and tacrolimus, which Nisha was also mentioning. This cyclosporin and tacrolimus, they interfere with the translocation of this nuclear factor of activated T cells, which is NFAT, to the nucleus. So it does not allow this NFAT to come to the nucleus, and that is how it does not allow this IL-2 the IL-2 transcription. Okay, so if NFAT cannot come to the nucleus, it will not be able to induce the IL-2 transcription, which is why the IL-2 mechanism, that IL-2 response by which the T cells can proliferate, that will be stopped. That is the role of cyclosporine. Next, coming to the CTLA4 FC fusion protein, which is the biletacept. What it does is that it binds to B7 and prevents the generation of this B7 CD28 co-stimulation complex. So it inhibits T cell proliferation by inhibiting this co-stimulation by the B7 CD28, right? Next, coming to the basiliximab, what it does is that it binds to this high affinity IL-2 receptor and does not allow the T cells to undergo IL-2 signaling, right? And another is sirolimus. Sirolimus, what it does, it interferes with this mTOR cascade, and therefore the T cells cannot differentiate, right? And the last one here is this azathioprine, which inhibits the replication and the proliferation of the activated T cells. So it stops the cell cycle, right? So you can see that these are the several different drugs which are used to inhibit the proliferation and the activation of the T cells, right? So any doubts here?
These are all in the recipient body.
These are all treated to the recipient before the transplantation. So that actually reduces any kind of T cell response in the body of the recipient. And then the transplantation is done, and also like during when the transplant is surviving, then also these immunosuppressive drugs are used for some time, so that there should not be any immune response.
Okay, all they give or some, like based on?
No, these are several different types of drugs, but I don't think all are given. Like of course some combination of some of the drugs maybe. Yes. Yes, sorry, what was your question? How dendritic cells only come along with the donor organ? No, all types of cells actually come. There can be multiple other antigen presenting cells also. But dendritic cells, as you know, that they can activate the T cells and the B cells, right? So therefore the role of dendritic cells is quite important in this case. Can we move on? Any other doubts from here? All right, then this is one important topic, that is graft-versus-host disease. And this is actually nothing but reverse of whatever we were discussing so far, the anti-graft response or the graft rejection. This is something which is actually the responsible T cells here are the T cells of the donor, right, and not the recipient. That is why it is the reverse of graft rejection. So what happens here is that when hematopoietic stem cells are transplanted, so in several cases of leukemia, in lymphoma, what we do is this bone marrow transplantation, right? So the hematopoietic stem cells, they are often transplanted to the donors. And while this kind of allogenic hematopoietic stem cells are transplanted, then these hematopoietic stem cell population, these also contain several mature and memory T cells. Okay, so these are also transplanted along with the hematopoietic stem cells. So now what happens, these T cells, they circulate in the blood and reach the secondary lymphoid tissues, Now these alloreactive cells, they interact with the dendritic cells and they proliferate. Alright. So these are what? These are the T cells of the donor and not the recipient. Okay. So what happens? The mature donor T cells, they recognize the tissues of the recipient as the foreign. So here the recipient is treated as the foreign. Okay, that is the difference between graft-versus-host disease and the graft rejection, right? And then it causes severe inflammatory disease in multiple tissues, but it particularly involves the skin, the intestines, the liver, and there can be rashes, there can be several patches on the skin. It can cause diarrhea, it can also cause liver dysfunction like that. And because the consequence of this graft-versus-host disease are particularly aggressive between the, so when there is a mismatch of MHC class 1 or class 2, that is why this HLA matching is very critical in case of HSC transplantation than solid organ transplantation, right? Because of this HSC transplant, so the T cells are not transplanted when the solid organs are transplanted. Are you getting my point? When the solid organs are transplanted, there might be presence of the antigen presenting cells, but not the T cells and the B cells. The T cells are only transplanted when there is a transplantation of these HSCs or hematopoietic stem cells. So when these hematopoietic stem cells are being transplanted, there has to be a very thorough checking of between the haplotypes of the MHC class 1 and MHC class 2, so that this kind of graft-versus-host disease, a severe inflammatory disease, cannot be generated in the recipient. Okay? And also remember that this graft-versus-host disease is very common in case of the differences between minor histocompatibility antigens as well. So which is why, as you understand again, that immunosuppression is very, very necessary in all these cases, right? Because even if there is MHC matching, the minor H antigens can be present, which can cause these kind of graft-versus-host diseases. Is that okay? Any questions? Now the last topic that I'm going to discuss today is the fetal tolerance. The tolerance of the fetus, and so that there is no immune response generated against the fetus. As Vandana was mentioning some time ago, that fetus also contain the MHC haplotypes from the mother as well as from the father. So here you can see that the mother and the father, they differ in the MHC haplotype, and therefore the fetus, when the fetus will be present inside the mother's womb, it will carry the mother's MHC as well as the father's MHC molecules. So now if there is an immune response generated against this fetus, then therefore the fetus will die. So there will be no pregnancy, right? There will be no, the mother will not be able to conceive. So therefore, this is an allograft, because you understand that this is also like a grafting, a new, this is like the transplantation, which is being tolerated repeatedly. So the fetus is tolerated repeatedly, and how is this being tolerated? So I will discuss this. First of all, there are actually, so this feto-maternal tolerance, this is there are several ways this feto-maternal tolerance is achieved. So there are multiple factors which are involved. So I'm going to discuss one by one. first one is MHC. So there is this outer layer of the placenta which is called the trophoblast, right? The trophoblast does not express MHC class 2 molecules and express only very low levels of MHC class 1 molecules. So MHCs are underexpressed on the trophoblast, right? That's the first point. Next, coming to the, so even if the MHC molecules are underexpressed, then also there can be the fetus can be attacked by the NK cells. So the NK cells, which are like the NK cells, are also very minimally expressed in the trophoblast layer, and the attack of the NK cells can be prevented by the expression of a polymorphic HLA class 1 molecule which is called HLA-G. HLA-G is a class 1 MHC molecule which protects the trophoblast from the attack of these NK cells. Okay, so actually what it does is that it does not allow the NK cells, the natural killer cells, to kill, right? So that is the second round of tolerance. The next is, which is one very important enzyme which is involved, that is the indolamine 2,3 dioxygenase, or IDO. So IDO is an enzyme which is expressed at a high level by the cells at the maternal and fetal interface. So at the interface of this mother's tissue as well as the fetal tissue, there is this expression of this enzyme, particular enzyme IDO. Okay, and what it does is that it inhibits the essential, or it depletes the essential amino acid which is tryptophan. So depletes tryptophan. Now, because of this depletion of tryptophan, the T cells, they are starved of tryptophan and they show this reduced response because of this reduction in tryptophan levels. And if this IDO is inhibited, as the scientists have observed in mice, if this IDO is inhibited, then it causes rapid rejection of the fetus, right? So this is a very important enzyme for the feto-maternal tolerance. Next, coming to the cytokine milieu. So the cytokine milieu at this interface of the mother's tissue with the fetal tissue, it also contributes to fetal tolerance. How? So the uterine epithelium as well as the trophoblast, so the mother's tissue as well as the fetal tissue, they secrete these anti-inflammatory cytokines which are TGF-beta and IL-10. Now this combination of these cytokines, they suppress the development of these effector T cells and favor the development of iTreg cells, which is the fifth point, iTreg cells. What are these iTreg cells? These are induced Treg cells. These are induced Treg cells. So what happens is that because of this induced Treg cells, these induced Treg cells, they suppress the response of any immune response against the fetus, right? So the cytokines as well as the iTreg cells, they help to suppress any particular immune response. Okay, and the last one is the chemokines. So the chemokines that attract the T cells to the antigens, these chemokines are, so they have reduced expression of the chemokines. Okay, so the stromal cells of the maternal uterine tissue, which directly interfaces the placenta, it appears to repress the local expression of these T cell attracting chemokines, right? So the maternal tissue, the maternal uterine tissue, it does not express these T cell attracting chemokines, so that the T cells cannot be attracted to this fetus, right? So these are the several ways by which this feto-maternal tolerance is achieved, and of course you can understand that it is absolutely important for the propagation of the species as well, so that there cannot be any generation of any particular immune response against the fetus, right? So these are the several ways this tolerance is achieved. Any doubt?
Last one is the paternal stromal cell, right? Reducing the chemokines.
Yes,
Paternal
the
or
stromal,
maternal?
the stromal cells of the uterus of the mother.
Oh, mother only.
Yeah, yeah. So, any doubts? If not, then we will directly quickly move on to the question answers for today. Please respond in the chat box. Yes. So the correct answer is the T cells. The graft rejection occurs due to the response to the T cells. Next question. Yes. So the components which inhibit the mother's T cells by nutrient depletion. So I'm mentioning here only one thing, that is inhibit the mother's T cells.
Tryptophan depletion.
Yes, by nutrient depletion, which is tryptophan depletion. The answer will be A. So all of you who have answered D, that holds true for the tolerance mechanisms at the maternal fetal interface. But the question is specific here. It is asking which is the component which inhibits the T cells by nutrient depletion, and that answer will be A, IDO. Okay, is that clear? All of you who were answering all of the above, that is true in the context of the tolerance mechanism, but not in the context of this particular question. All right, moving on to the next question. What causes this hyperacute graft rejection? Okay. Right. So the correct answer is D. Antibodies against the donor blood group. All right. Fourth question. Yes. The correct answer is A, the autoimmune regulator gene. And the last question for today. The correct answer is the minor H antigens, the polymorphic cellular proteins which caused several problems in the transplantation process. That is the minor H antigens. Okay, so that's all for today. Thank you all for joining. Let me know if you still have any doubts. And Sandeep, I will get back to you. I will respond to your email. Just give me
Yeah, yeah,
some more time.
yeah,
Okay.
yeah. Okay, thank you, Surasree,
Thank you.
and Shubho Vijaya.
Shubho Vijaya to you too. All right. Thank you so much for joining today. Next Tuesday will be our last session. So please join all of you and happy learning. Okay. Bye.